Everyone, my name is Maddie, and I've been catching bats in Iowa for the past few summers with my parents, who are also field biologists. Having worked so closely with bats, their conservation is very important to me, so I decided to make this a key aspect of my senior thesis. Like I said, I'm from Iowa, and it used to be mostly tallgrass prairie and oak woodland, but has since been converted mostly to agriculture. Crops don't look like they would support a lot of the native wildlife, but crops attract a lot of insect pests, which provide a lot of prey for bats. Bats provide crucial ecosystem services in this area, saving the American agricultural industry $25 billion a year in pest control services. There have been 9 species documented in Dubuque County. They're all members of the same taxonomic family, Vespertilionidae. 4 of these species migrate through the county, while the other 5 hibernate over winter. This species, Myotis septentrionalis, is this a pointer? Yeah. Myotis septentrionalis is federally threatened, and Myotis sodalis is federally endangered, though it hasn't been detected in the county for several decades. Perimyotis was also recently listed as being under review for being federally listed. All of these bat populations are endangered due to 4 big threats. Climate change is affecting all species right now. Habitat fragmentation also affects all species because it impacts the prey composition and roost availability in the area. Wind turbines kill about 100,000 bats every year and disproportionately affects migratory bats. The last big threat is called white nose syndrome. It's caused by a fungal pathogen that was introduced to New York in 2006 and has been moving west ever since. This disproportionately affects small hibernating bats, and in affected colonies, it can kill up to 99% of the individuals in a single year. With all of these threats to bat populations in mind, I decided the goal of my research project was to inform management strategies that benefit the conservation of bats where I'm from. To do this, I asked how environmental variables influence bat detection, occupancy, and activity in Dubuque County. I defined detection as if a bat will be recorded if it is present. Occupancy was if a species is present in the area, and activity was the number of calls in a night. I identified 22 sites throughout the county that represented areas with different local environments. To find how landscape variables predicted occupancy in these areas, I used ArcGIS to calculate the distance to 6 types of land use listed here. And I also calculated the land use composition within a 500-meter buffer region of my sample sites. Like I said before, these bats echolocate, and I used bat detectors. Here you can see my setup. And I recorded bat calls from 1 hour before sunset to 1 hour after sunrise. I recorded each site on 4 nights throughout the summer for a total of 88 sampling nights. On each sampling night, I recorded weather data like temperature and humidity to find out how these variables might impact bat detection. After I gathered all of my acoustic data, I ran it through kaleidoscope analysis software. It takes a sound file and spits out this image, which it can record as a different species, since each species has a unique echolocation signature. Here are the results of my acoustic data throughout the summer. Most notably, you can see we identified the endangered species over 400 times. And in general, these species varied a lot in the amount of activity and the number of detections over the summer. Unfortunately, my occupancy models don't produce results with a low enough standard error if the bat is detected too frequently or if it's too rare, so I only have models for these 4 species highlighted in green. However, all of the bat species had enough activity for me to run activity regressions in R. Here's an example of my workflow for one of the species, the little brown bat, Myotis leucippus. My first step was to determine if this species was present at each site on each night. So here's some sample data. You can see at sites D and V that it was present on some nights but not others. This means that there's imperfect detection. To find if any weather variables impact detection, I ran models using these variables. For this species, the null model performed best, meaning that none of the variables predicted detection well. So I moved on and determined whether landscape variables impacted bat occupancy. For this species, the model that performed best included one variable, forest cover. In order to visualize how this variable impacted bat occupancy, I plotted the variable. Here's an example where we have 2 species whose occupancy models included percent forest, though forest cover predicted occupancy differently for this species. Myotis oesophagus preferred to occupy areas with more forest, while Lasiurus borealis preferred to occupy areas with less forest. We can repeat this process for the detection, occupancy, and activity of variables to see the sensitivity of the species to these factors. When we synthesize the data, we get a big chart that looks like this, and it's a lot of data to digest, so we're going to go through it by species group and relate our findings to bat ecology. First, Aptescus fuscus is in a group all by itself because it's the only hibernating bat in the area that's really large. Because it's large, it specializes on larger prey like beetles and agricultural pests, and we can see this reflected in the model. They're more likely to occupy areas with more agricultural cover, and I believe this is most likely because of the prey availability in the area. Our next species group is the Myotis bats. These bats are a lot smaller than Aptescus, and we can see this reflected in the models because they're more likely to be in areas with more forest cover and wetlands, which support an insect community that is smaller like mosquitoes. The next species group includes Perimyotis. This is the smallest bat of the group, weighing only a few grams. It's really cute. And we can see this reflected in the model because it also occupies areas with more forest cover like the Myotis bats, because they also eat really small prey. Interestingly, we can see that it's more likely to be detected on nights that are warmer. This might be because it's too small to support itself energetically when it's foraging, or that its prey are too small and that they're not active when it's cold out. Lastly, we have the migratory bats, these four at the bottom in yellow. Migratory bats have long, thin wings that are good for flying fast and far, but they're not great for navigating areas with a lot of vegetation. We can see this reflected in the model because they tend to avoid areas with forest cover, and they're more active further from forests. Interestingly, for this species, they're more likely to be detected when there's more moonlight. This could mean that the moon is important to help them navigate. Since they are migratory, they could use visual cues to help them navigate the landscape. So what do we do with all of this data? First, we need to monitor the current species populations, both with more acoustic recording and by netting these bats. Especially for the endangered species Myotis sodalis, netting the bats and physically documenting them is a really important step into protecting them federally. We also need to monitor disease progression, and if some species are doing worse than others, we need to readjust our management priorities. If we see that bat populations are declining, like we have with the Myotis bats in recent years, we need to take action by protecting the preferred habitats, and the data that I just showed you can inform those management strategies. If we're able to protect those areas, we can maintain a healthy, diverse bat community and maintain our ecosystem benefits. I've learned a lot from this project, mainly that occupancy models can help determine habitat preferences for bats in Dubuque County. Each bat species has unique ecology and thus demands unique management strategies. And lastly, I'd like to emphasize that there needs to be more effort to net the endangered bat Myotis sodalis in the area in order to protect them federally. And I want to thank my lab group, the Beisinger Lab, my parents, my environmental science cohort, Cody, and Sam Gunn. Thank you all so much for being here.